Hey everyone, time to talk about viruses. So this lesson doesn't have a lot in it, but it can be a lot to chew on, okay? Because it's kind of new for you guys. So in BIO112, you guys probably talked about viruses as just molecules, which they are. Like you can see here on the screen, they're just a bunch of molecules. You can see a protein coding. So here the purple molecules around the green string. The green string would be the DNA. You see another shape here when you have a bacteriophage. We'll talk about it in a second, but basically it's all proteins, and inside that little crystal there is DNA. You can have something that looks really similar to COVID when you have an envelope, some spike proteins, and inside that envelope there is just some DNA floating around. So you have some protein coding that is a little bit more different. So this is HIV kinda. But the basic thing to remember about viruses is they are just a protein coding. There is no cell membrane, and inside there is DNA or RNA, and that's it. That's really it. And some proteins inside, but that's all. That's all what a virus is. So viruses, you see, they come in different shapes, and usually that's how we classify them: shape and and the way they do things. Which cell they infest and all. But do realize that viruses don't really have a metabolism, so it's not like they're doing anything. It's just like this, and then when they infect a cell, that cell does metabolism, it does reproduction, does a lot of things for that virus, which means that viruses kind of break down the boundaries of being alive, because what you remember from the five characteristics of life, one of them is develop, growth, reproduce. Metabolism is one, so homeostasis, and viruses do none of that, and they're not even made of cells because they don't have a phospholipid bilayer surrounding themselves. So that's a really weird thing, but it is viruses for you. So let's take a look at what we mean now when if we take one virus, the bacteriophage here that you're seeing, and you see a real picture of it, which is colored by artificially, but it's exactly what they look like if this has been taken in real life with an electron microscope, transmission electron microscope, and you can see that virus here is injecting the molecule of DNA, the green string here, as is that one and this one, into the cell. And so they infect specific host cells. They don't infect everything. They have a target, and that target is really specific, and it's because they need to be able to attach themselves on top of a cell. Notice also the difference in size between the virus and the cell. So the host cells, that's which cells are infected by that virus. So for example, the HIV, it's white blood cells, right? But white blood cells are present in more than one organism. That's why we have something called the host range. And that host range, that would be all the organisms that are infected by the virus. So if you have a host range, it's all the organisms that the virus can infect, and then the host cells is the type of cells that the virus can infect. Because you understand that you have white blood cells in other organisms than just humans, for example. And in fact, the host range for the HIV virus, which will be this one here, is more than just humans. Okay, so now when I say that they're really specific to the cells, because of the first step in their process, and that first step is attachment. So you see, it needs to bind to a surface protein, surface receptor, and that receptor here matches the protein code of the virus. So the virus, once again, is just that coding with DNA inside. Ignore the fact that there's a cellular membrane here and some cell wall. It's more complicated than that. It's not cellular membrane. This is just protein coding. So when it attaches, it can only attach to its host cell, right? Once it has attached, now it's ready to penetrate, and the penetration is when the nucleic acid, the viral nucleic acid, in this case DNA, is just released inside of a cell. So attachment is one, penetration is second. The third one is synthesis. So in synthesis, that's when the viral DNA is used for two things. First, transcription, when you make RNA, and then translation, when it makes more of a protein coding to make more viral particles. And viral DNA replicates. So this is a like we'll call that a bacteriophage virus in that case, because this is simply a bacteria. You see no organelles inside the cell. So DNA inside of a cell can right away be used by ribosome, can be used by all the replication machinery, and make more viruses, right? So that's pretty straightforward in that case. So you have after penetration synthesis, and after that assembly when new viral protein particles are made, and then there's release. So those 5 steps here, that are attachment, penetration, synthesis, assembly, and release, you must know them. You must know them in order. You must know them backwards, disorder, all of it. So you just have to learn it. But you don't learn it as in a vacuum without understanding it. That's going to make it so much harder to learn the principles. You want to understand how a virus works. Think about it. It's just a viral part, a DNA surrounded by proteins. So when it's trying to infect a cell, its host cells, it has to just first attach to it. Once it's attached, it has to get in. Once it's in, then it can be replicated and make new particles of viral particles, because those viruses don't do their own replication, right? So now that you're there, you understand that this is ever so slightly closer to make a full infestation going on, and for that you need to release said viral particle into the body or the environment, right? That makes sense? So really understanding the process will help you, but you have to learn it. And you have to learn some subtleties of the viral processes. One subtlety is when your virus is based on RNA. So why would that be a problem when it's based on RNA? Well, let's imagine back here, and we have attachment, penetration, and synthesis. If it's not DNA that's injected, but RNA, directly here, whenever RNA is used by ribosome to make protein, first of all, you limit it by the quantity of RNA you put in there, because there is no such thing as RNA polymerase, like RNA replication, sorry. There is no such thing as RNA replication in cells. They duplicate DNA. So all of a sudden, you're bound to just use the RNA you have. So not only can it get damaged, but it's limited in the number of particles you can make, right? Because also that RNA, if you just inject RNA, will have to be used to make the proteins and then sent out. So the virus doesn't really multiply. It's just stuck with the number of RNA it has, right? So RNA viruses have something really special that's called reverse transcriptase. So the reverse transcriptase is an enzyme that is present in the viral particle, which after attachment and penetration will then be used to make that RNA-DNA hybrid that will then be incorporated into the nucleus. And it's in the process of making the DNA hybrid that it gets into the nucleus. But then once it's in there, you can start making transcription translation. And that allows RNA-based virus to get transcription. Transcription is powerful because it makes a ton of copies of the viral RNA. And so now you can do a lot of translation inside of a body. You're not limited to just the RNA that you injected the cell with. And then you also make more copies of RNA, viral RNA. So all of that is then released, and so you get an infection that is growing, right? So it's really important to understand those differences, the subtleties, but it's still attachment, penetration, synthesis, assembly, release, right? There is only the subtlety of a reverse transcriptase. That's typical of the HIV virus. Notice that this also allows more complex organism to be infected, because now you can infect cells that have organelles. Before, if you just release DNA into the cytoplasm, if you just release DNA here in a eukaryote with a nucleus, nuclear envelope, the DNA is never going to get to the transcription machine. So here in this case, the key is that viral DNA is right away able to be replicated. Whereas in a eukaryote, you don't have a replication machinery in the cytoplasm. So if you just inject DNA, you're going to have issues, right? So as a rule of thumb, DNA viruses tend to infect prokaryotes, and RNA viruses tend to infect eukaryotes. It's a rule. Doesn't mean there are no exceptions. There are many exceptions. But that's how you use your understanding of BIO112, right? To get a little bit better at understanding what's happening here. So really, that lesson here relies on you understanding really deeply transcription and translation. So that could be a good moment for you to revise those, to go back to your notes. Because you notice that we just said transcription, translation, we don't explain any of that. And DNA replication, same thing, we just write it, but you expect it to know what the machinery is and all of that. So yeah, that's the subtlety here. And finally, as you notice, there's a release here, and we'll learn about bacteria that this could be a problem that could lead to the death of a bacteria. So what happens if you kill the host cell, bacteria of a eukaryotic cell? Well, we are in that case, we are in what we call a lytic pathway. So the lytic pathway here is when your virus infects, right here, and then the viral DNA is released, so attachment, penetration, and then here you have synthesis, assembly, right, and release. So all of that happens, and the release kills the bacteria. So you notice the bacteria dies because all those bacteriophage are released. It pierces the membrane in too many places, right? So that's lytic pathway, and that's lethal. Lytic is lethal, so there's a LT, lethal, lytic, it kills the host. However, if that was happening every time, well, you run out of host, right? And in more complex hosts, it could be good, or even in prokaryote, it could be good to make sure you stay in there for a while. So how does that work? In that case, after attachment penetration, you don't get to synthesis. What you do is you just stay dormant. You remain in the DNA, and you don't get expressed for a while. And chromosome replicates, and you get more and more copies, and so now you have more and more cells that are infected. That is typical of some viruses, such as herpes in human. This is a prokaryote here, but herpes in human stays in you, COVID stays in you as well, and that's the lysogenic pathway. That is a latent state in which the virus just replicates with the DNA of the host. Now, this is really helpful, and it can switch back to lytic at a moment's notice. And usually viruses do that for herpes, for example. You all know about cold sores. Cold sores come from when you're stressed, when your immune system is compromised. So whenever you are stressors that take some energy and resources, and then now all of a sudden you cannot prevent the expression of a virus, which means that the virus, although it's in lysogenic pathway, every now and then tries to go into lytic, but your immune system is capable of keeping it under check. But once in a while you're too stressed, too tired, and it cannot, and so there is an outburst. So that's when you get a cold sore. For COVID, that's when you can get another onset of a disease. So we all kind of carrying those viruses in our DNA, like in our cells. This is a little bit more complex for eukaryotes compared to prokaryotes. As you can see here, prokaryotes don't have organelles, so if you want to think about the lysogenic pathway, in here you can understand it. The DNA, it's as it's represented here, the DNA gets incorporated. In there, you would have reverse transcriptase get incorporated to your DNA, and it doesn't get transcripted right away. Doesn't get right away used. So, you know, like slight subtleties based on which host host range we're talking about. But those are the two pathways. So that's the lesson on virus. For a full understanding of what's happening, it's really important that you first focus on learning the 5 steps. So assembly, attachment, penetration, synthesis, and assembly, release, right? Learn those steps, get them under control, then understand what is a host cell versus a host range, and finally distinguish between viral DNA or viral RNA with a reverse transcriptase, and I should say now finally the subtleties of lytic versus lysogenic pathway. Those, to make sure you learn all of that, it's great to quiz yourself, it's great to try to explain it to someone. So if you can try to explain that to someone and see if you are capable of drawing the whole thing of what's happening, the 5 steps, if you're capable to explaining the difference between lytic and lysogenic, or just draw on a whiteboard. I really encourage you to draw on this lesson. Those are like perfect example when you can draw. You don't have to draw all the details, but if you can draw what's happening and put the main arrows like replication of DNA, transcription, translation, reverse transcriptase, all of those, it really will help you. So that's the lesson on viruses. That's mainly what you need to focus on and learn for this unit. And it comes with drawing and repetition. So now it's your turn. Go for it. I'll see you in the next one.